Welcome to Biology Made Easy. Today in this video we'll discuss polygenic inheritance. So as you know that the phenotypic traits are mainly of two types. It may be qualitative or it may be quantitative. Qualitative or quantitative. The phenotypes. So what is qualitative phenotype? As we have seen that in the Mendel's classical experiments, there are two contrasting characters. For one gene, there are two alleles, two contrasting characters. And a single gene has one dominant allele and one recessive allele. And if that gene is present either in single number or both are present in homozygous condition, the effect is same. For example, suppose we will take this genotype, that means it is tall. If we take it homozygous this condition, this is also tall. That means there is no difference in the phenotype even if the same gene is present in single number or in two numbers. Here a single gene controls everything. So this type of inheritance is known as monogenic. Monogenic. Mono means one. That means a single gene controls all these characters, and that's the example of all Mendelian characters. But if we look around us, if we look around the world, we'll find that all the characters are not contrasting. Suppose you take the case of human beings. In case of human beings, do you think that the height of the human beings is either tall or dwarf? No. There are so many intermediate types are there. Then think of the intelligence of human beings. Are the human beings completely intelligent or idiots? No. So many intermediates are there. Similarly, take the case of hair color. Hair color also it is not contrasting. So many sets of colors are present. You take the facial forms. Then again you will find different facial forms. So these characters, I have given you the examples of human beings. These are not contrasting characters. Different sets of the characters are present. So such characters are usually known as quantitative, and they are regulated by several genes. Instead of a single gene, as in case of qualitative one, in quantitative inheritance, so many genes, or you can tell two or more genes, they control a particular character. So such are either known as quantitative inheritance, or it is known as polygenic, or it is known as multiple, or also you can call it as cumulative. And all these are usually governed by two or more genes instead of one. So when two or more genes will act on a particular phenotype, the net effect will be equal to effect of gene 1 plus effect of gene 2, effect of gene 3, and so on. And again, in each case, it is only the dominant allele that is responsible. That means, suppose there are two genes A and another B, suppose another C. That means it is only the dominant alleles, they are responsible. The recessive alleles are not responsible for the characters. That means the dominant alleles, they contribute to the character, while these are non-contributing. Then let us take one example of this polygenic inheritance so that our understanding will be still clear. The best example for this polygenic inheritance is human skin color. And this human skin color that was first studied by Davenport brothers Davenport brothers, they have studied it first. So human skin color, there are different shades, different grades of colors are there. So the human skin color is usually controlled by 3 genes, 6 alleles, and they are present in different chromosomes. And it is only the dominant genes, they will control the whole thing. So you will take the human skin color as an example of this polygenic inheritance. The human skin color is controlled by 3 genes, A, B, C. So only when they are present in the dominant form, they will express the phenotype. Their recessives will not express any phenotype. Now these are present in different chromosomes, but their effect is same. That is determination of the skin color. Usually for the skin color of human beings, the pigment is melanin. The melanin pigment. Now the greater the amount of melanin, the darker the person is. And the less the amount of melanin, the person will be white. That means whether a person will be black or white, that depends upon how much of melanin is present in the skin. So, if all these 3 genes are responsible for the human skin color, now let us 2 examples. Suppose let us take a very black person, like a Negro. So in that case, it will be, the genotype will be AA, BB, CC. That means very dark color. If such a person will marry with another white person having only recessive genes, that means very white, what will happen? The F1 will be heterozygous, capital A small a, capital B small b, capital C small c. That means here 6 dominant genes are there, here recessive are there, here 3 dominant genes are there. That means this is the intermediate color between very dark and very white, and this is known as mulatto color. It is intermediate type. Now, if 2 such persons will marry, what will happen? Now, this is the F1 generation. You will study the F2 generation. In the F2 generation, suppose 2 such persons will marry. Then how many types of gametes they will form? They will form 8 types of gametes. What are the 8 types? You see, what are the different 8 types? One is capital A, capital B, capital C. Then 2 dominant genes. Capital A, capital B, small c. Then capital A, small b, capital C. Then small a, capital B, capital C. In this, here 3 dominant are there. In all these cases, 2 dominant genes are there. None, single dominant gene. Single, capital A, B, C. Then small a, capital B, small c. Then small a, small b, capital C. In all these cases, only 1 dominant gene is there. And then all are recessive. You see how many types are there? 8 types are there. In this side also, this same 8 types will be there. A, B, C, then A, B, C, then A, small b, capital C, then small a, capital B, capital C, then capital A, small b, small c, then small a, capital B, small c, then small a, small b, capital C, then A, B, C. 1, 2, 3, 4, 5, 6, 7, 8. Then you see, here there are 3 dominant genes, here there are 2 dominant genes, here 2 dominant genes, 2 dominant genes, here 1 dominant, here 1 dominant, here 1 dominant, that is no dominant. In this case also, we will find here 3 dominant genes are there, here 2 dominant, here 2 dominant genes, 2 dominant genes, here 1, here 1, here 1, here nothing. Now, if the checkerboard will be drawn, the checkerboard will be drawn, we will not write all those things because it will take around 5 minutes, not necessary, but simply I am explaining it to you. So, you see, in this side there are 3 dominant genes, in this side there are 3 dominant genes. That means here there will be 6 dominant genes. 6 dominant genes. And at no other place there will be 6 dominant genes. That means this will be very dark. If we come to this, from this side 3 dominant, here 2 dominant. That means 5 dominant genes. Here 3, here 2, again 5. Here 3, here 2, again 5. Here 3, here 1, so only 4. Here also 4. Here also 4. And here only these 3 are there, because this side are not there. Similarly, if you go to this side, here 3, here 2, 5. Likewise. And in the last one, where this side also ABC and that side also ABC, in the last one, we will get double A, double B, double C in the last one, where there is not a single dominant gene is there. That means this will be very light. Now, if we analyze all these things, what we'll get? We'll get very dark, only one, this one, very dark. Why? Here we will get 6 dominant genes. 6 dominant genes. Then next is dark, only dark. Dark, how many will get? We will get 6, where there will be 5 dominant genes. Then we will get fairly dark, and we will get 15, where there will be 4 dominant genes. Then in the intermediate type, it is mulatto or intermediate type. Here we will get 20, and here is only 3 dominant genes. Then fairly light will get again 15, and here 2 dominant genes are there. Then light, again will get 6, and here 1 dominant gene is there. And very light, only 1 is there, and no dominant genes. That means here 8 into 8. So the number can be classified into this. That is very dark, having 6 dominant genes, 1, dark 6, fairly dark 15, where there will be only 4 dominant genes, and mulatto or intermediate type will be 20, where we will get 3 genes, fairly light 2 genes, and light 6, and very light, there is no dominant gene. That means the phenotypic expression is designated as 2n plus 1, where n is equal to the number of genes. That means 2n, here n is equal to 3, plus 1 means 7. So how many types of phenotypes are there? You see 7 types, 1, 2, 3, 4, 5, 6, 7, 7 types. Then what will be this is the phenotype. Now if you come to the genotype, how many genotypes? 3 to the power n. And here 3 to the power is 3 to the power 3, means 27. That means 27 types of genotypes and 7 types of phenotypes will be usually obtained. Now, if we will draw the entire thing in the form of a histogram, if this will be drawn in the form of a histogram, now see at the center, this will be 20, where there will be 3 dominant genes. That means this mulatto. Then on this side there will be 15, and on this side also there will be 15, and this side there will be 4 dominant genes, this side there will be 2 dominant genes, because this side will be fairly dark. When 4 are there, fairly dark, and when 2 are there, fairly light. Then next, here we will get 6, and here also we will get 6, and here we will get 5 dominants, and here we will get single dominant, because this will be light. Then at the 2 extreme ends, there is a num, where in this extreme end the number is 1. Here the number is 1, because this is only 1, and this is also 1. This is also 1. So this is 1, but here you will get 6 dominant genes. 6 dominant. That means when 6 dominant genes, it is only 1 in number. When no dominant gene, also it is 1 in number. That means initially we have taken the parental characters, very dark and very light. The parental characters are appearing very less, while the intermediate types are more in number. That is observable from this histogram. The intermediates are more in number, while the extremes are very less in number. So this we are obtaining from this histogram. So besides this experiment by Davenport brothers in the human skin color, there are also other experiments done by other scientists. For example, if we take the case of Carl Reuter. Carl Reuter, he has studied the height of tobacco plant, that how it is polygenic. Similarly, the Galton, he has studied the height as well as intelligence of human beings. Similarly, Nelson and Elle, they have studied the kernel color of wheat. All these are examples of polygenic inheritance. Now one question comes to mind. How several genes are regulating a single character? Polygenic inheritance means several genes, they are regulating a single character. How is it possible? What is the cause behind it? So we will discuss the cause behind it. There are 3 probable causes. There are 3 probable causes. Number one is mutation. That means the mutation has same effect. And number two, we can tell that it is due to duplication of chromosomal parts. Or the third one, it may be due to polyploidy. Because in polyploidy, the same chromosome is double, triple like that. So these are usually 3 probabilities or 3 causes why this polygenic inheritance occurs. So you have a now good idea about the polygenic inheritance along with the example. Now let us discuss questions. So one question is grain color in wheat is determined by three genes. A, B and C. If a cross is made between capital A, capital B, capital C with small a, small b, small b, small c, small c, then in F2 generation, the parental types will be first choice half, second choice 1/3, third choice less than 5%, and fourth choice none of the above. So this question was once asked in AIMS. Now come to the second question. Only the dominant allele act as contributing alleles in case of first choice multiple alleles, second choice blending inheritance, third choice codominance, and fourth choice multiple factor inheritance. So these are the two questions. I hope you will be able to answer both the questions. Now you have a very clear idea regarding the polygenic inheritance. Thank you very much for watching the video. God bless you and have a good day.